Hello students, in today's class we will discuss very interesting topic, that is molecular basis of inheritance. In the previous chapter, that is principles of inheritance and variations, you have studied the patterns of inheritance and basis of genetics or their genetic basis. So in this chapter you will study the structure and functions of genetic material. So you already studied in the topic genetics, a character in an individual is controlled by a pair of genes or factors. The question arises, what are these genes or factors? They are nothing but a fragment of DNA. Then what is DNA? It is a type of genetic material. Then what is genetic material? The substances or molecules which stores and carries the biological information in the coded form and transfer the genetic information or biological information from one generation to the another generation. And you remember one thing, these molecules are expressed in the form of proteins. The genetic material which stores a biological information in the form of coded form, and those biological information are expressed in the form of proteins. So when you talk about genetic material, DNA and RNA act as a genetic material. Why DNA and RNA are called as a genetic material? It should possess some properties or characteristic features. The first characteristic feature of genetic material is it should transmit biological information or genetic information from one generation to the other generation. The second property, it should be stable and replicate. That means genetic material should exhibit stable mutations, and those stable mutations which are inheritable must be transmitted to the next generation. Why I use the term stable? The stable mutations leads to useful variations, and those useful variations helps in evolution. Is that clear to you? The second one, why should genetic material replicate? In the previous point we discussed information transmitted from one generation to the other generation. The genetic information is stored within the DNA or genetic material. At the time of cell division, what happens? The DNA or genetic material doubles or replicates. After that, cell division takes place, so that the information transmits from one generation to the other generation. The next one, the information for development and structure and metabolism of the cells. And the fourth property is it should undergo mutations which are required for evolution or which are required for variations and origin of new species. Come back to the genetic material or nucleic acids. Nucleic acids are the polymers of nucleotides, or we call it as a macromolecules. These macromolecules were first discovered by Frederick Michel in the year 1869 from the nuclei of pus cells, and he named it as nuclein. Later on, the student of Frederick, Altman, in the year 1889, he renamed nuclein as nucleic acids because of acidic properties. According to Altman, Frederick Michel, there are two types of nucleic acids. One is DNA, another one is RNA. Deoxyribose nucleic acid and ribose nucleic acids. The DNA, it's a polymer of deoxyribonucleotides. That means nucleotides are the building blocks of nucleic acids. In case of DNA is a polymer of deoxyribose nucleic acids, nucleotides, whereas RNA is a polymer of ribonucleotides. That means the nucleotides are the building blocks of nucleic acids. Nucleic acids, or they are the macromolecule found in all living organisms. They are found in plants, they are found in animals, they are found in prokaryotes, they are found in viruses. In most of the living organisms, DNA acts as a genetic material, whereas in only few living organisms, for example few viruses, RNA acts as a genetic material. RNA acts as a genetic material. Come back to the occurrence. So you already know that in case of prokaryotic cells, DNA is circular, embedded within the cytoplasm, because in case of prokaryotes, nucleus is absent. That's why it is embedded within the cytoplasm. And such type of DNA is called naked DNA. Naked DNA. Whereas in case of eukaryotes, DNA is found in nucleus and in some organelles, cell organelles. The major amount of DNA is restricted to the nucleus, and very negligible amount of DNA is found in plastids and mitochondria. Okay, so plastids and mitochondria. The negligible amount of DNA is found in the cell organelles like mitochondria and chloroplast. Such DNA is called extranuclear DNA. Whereas the major amount of DNA is confined to the nucleus, such DNA is called nuclear DNA. So you remember one point. In case of prokaryotes, it is naked DNA. Why? Because it is embedded within the cytoplasm. Whereas in case of eukaryotes, you find two DNA. Nuclear DNA, it is restricted to the nucleus. Extranuclear DNA, it is restricted in some cell organelles such as plastids and mitochondria. When you talk about DNA, okay, already I told you it is a polymer of deoxyribonucleotides. And nucleotides are the building blocks of nucleic acid. And each nucleotide is made up of three components. The first one is pentose sugar, the second one is phosphoric acid, and third one is nitrogen bases. I'll repeat once again. The first component is pentose sugar, the second one is phosphoric acid, and third one is nitrogen bases. So come back to the first component of nucleotide, sugar, pentose sugar. A 5-carbon sugar ಅಂತ ಹೇಳ್ತೀವಿ. So in case of DNA, you find deoxyribose sugar. Deoxyribose sugar. Okay, 5-carbon sugar ಅಂತ ಹೇಳ್ತೀವಿ ಅಥವಾ pentose sugar. So chemical formula of deoxyribose sugar is C5H10O4. When you look at the structure of deoxyribose sugar, the 2 carbon carries only hydrogen atom instead of hydroxyl group. Look at here, this is first carbon and this is second carbon. One oxygen atom is missing in case of deoxyribose sugar. Okay, deoxyribose sugar. Hence, it has one oxygen atom less than the ribose from which it is derived. That means deoxyribose sugar is derived from ribose sugar. The only difference is one oxygen atom is missing in case of deoxyribose sugar. When you look at the structure of ribose sugar, look at here, this is first carbon position and this is second carbon, so oxygen atom is present. Okay, so this is about the first component. We'll move to the second component, phosphoric acid or phosphate group. The phosphate group is a derivative of phosphoric acid. And what is this? A phosphoric acid is an orthophosphoric acid with a molecular formula H3PO4. That means it consists of what? 3 functional OH group, okay, hydroxyl group, and 1 oxygen atom. And I told you the nucleic acids are acidic in nature. Why? Because of phosphate group. Okay, we'll move to the, and it is symbolically represented as capital P. We'll move to the third component, nitrogenous bases or nucleobases. The first question arises, what are these nitrogen bases? So you already studied the benzene ring. Okay, carbon hydrogen atoms. When you talk about nitrogen bases, these are cyclical nitrogen containing compounds in here. The cyclical nitrogen containing compounds, and these bases are derived from 6-membered aromatic benzene ring. So when you look at the structure, normally in case of benzene ring you find a CH. So in some position, instead of carbon atom you find nitrogen. There are 2 types of nitrogen bases or nucleobases. The first one is purines and second one is pyrimidines. So these are the 2 types of nitrogen bases, purines and pyrimidines. We'll move to the first one, purines. So purines are the type of nitrogen bases where nitrogen atom is at 1, 3, 7 and 9th position. So they are double ring structures. Purines are double ring structures with a nitrogen atom at 1, 3, 7, 9 position. When you look at the structure of purines, so look at here, nitrogen atom is present at first position, third position, 7th position and 9th position. Okay. There are 2 types of purines. The one is adenine and the second one is guanine. So There are two types of purines. One is adenine, second one is guanine. Come back to the second type of nitrogen bases, that is pyrimidines. The pyrimidines are single ring structures, six-membered aromatic ring with nitrogen atom at 1 and 3rd position. We'll recall the purines. In case of purines, nitrogen atom is found in 1st, 3rd and 7th and 9th position. In case of pyrimidine, nitrogen atom is at 1st and 3rd position. There are two types of pyrimidines. One is cytosine, the second one is thymine. Cytosine and thymine. Look at here. One of the difference between DNA and RNA is DNA is made up of deoxyribonucleotides, whereas RNA is made up of ribonucleotides. In case of DNA, the nitrogen bases are adenine, guanine, cytosine and thymine. In case of RNA, the nitrogen bases are adenine, guanine, cytosine and uracil. So instead of thymine, you find uracil in case of RNA. Okay. So when you compare pyrimidines and purines, or purines and pyrimidines, the purines are larger than the pyrimidines. Purines are larger than the pyrimidines. When you talk about nucleotide, so nucleotides are the building blocks of nucleic acid. How these nucleotides are formed? Look at here. So we'll discuss nucleosides, nucleotides and polynucleotides. Okay. The first one is nucleoside. When nitrogen bases combines with pentose sugar to form nucleoside. So already know that in case of DNA, adenine, guanine, cytosine and thymine. So all these four nitrogen bases combines with pentose sugar. In case of DNA, the pentose sugar is deoxyribose sugar to form respective nucleoside. That means 4 deoxyribonucleosides are formed. Okay. Look at here. The nitrogen base combines with pentose sugar to form nucleoside. So in between the nitrogen base and pentose sugar, a bond formation takes place. That bond we called as a N-glycosidic bond. N-glycosidic bond. Okay. So bond which is formed between nitrogen base and sugar. The first one, the purine or pyrimidine base. This is purine. Combines with pentose sugar. Okay. So in between purine and pentose sugar, or nitrogen base and pentose sugar, a bond formation takes place, and that bond we called as a N-glycosidic bond. You remember one thing, if it is purine, if nitrogen base is purine, the 1,9,1,9 glycosidic bond formation takes place. If it is pyrimidine, 1,1 glycosidic bond formation takes place. Okay. So 4 types of nucleosides. Adenine, okay, combines with deoxyribose sugar, to form deoxyadenosine. So this is deoxyadenosine, and the type of bond formation between sugar and nitrogen base is 1,9,1,9 N glycosidic bond. N glycosidic bond. The second one, guanine combines with deoxyribose sugar to form deoxyguanosine. Deoxyguanosine. So again, the nitrogen base is purine. Again, the bond formation takes place is 1,9 N glycosidic bond formation takes place. Then we'll move to the third one. Thymine combines with nitrogen base, pentose sugar, deoxyribose sugar to form deoxythymidine. Look at here. Here the bond formation takes place. 1,1 N glycosidic bond formation takes place. 1,1 N glycosidic bond formation takes place. Why? Because thymine is pyrimidine. And fourth one, cytosine combines with deoxyribose sugar to form deoxycytidine. Deoxycytidine. Okay. So 4 nucleosides are formed, and these nucleosides combines with the phosphate group. Okay. Combines with phosphate group are linked to phosphoric acid to form nucleotide. And you already know that in between nitrogen base and pentose sugar, a bond formation takes place, and that bond we called as a N glycosidic bond. And in between nucleoside and phosphoric acid, a bond formation takes place, and that bond we called as a phosphoester bond. Phosphoester bond, here defined, phosphoester bond. Okay. 2 such bonds takes place. Okay. One in between nucleoside and phosphate group. So another phosphoester bond takes place in between 2 nucleotides. In between 2 nucleosides. Suppose this is a 1 nucleotide, in between another nucleotide. This is just imagine here. Okay, one more nucleotide is present. In between these 2 nucleotides, one more bond formation takes place. That bond is also phosphoester bond. The phosphate and 3 carbon and 5 carbon of sugar of successive nucleotides. Okay, 2 phosphoester bond formation takes place. Nucleotide is a monophosphate. Monophosphate and third. Look at here. The first one, nucleotides are the monomers of polynucleotides. 4 types of nucleotides are formed, and they are the building blocks of nucleic acids. Nucleoside combines with phosphate group to form nucleotides. Let us see one by one. Deoxyadenosine combines with phosphate group, monophosphate, to form deoxyadenosine monophosphate. Deoxyadenosine monophosphate. What is this? It is a nucleotide. So DAMP, or it is called deoxyadenalic acid. Deoxyadenalic acid. So this is the first nucleotide, that is a deoxyadenalic acid or DAMP. The second one, deoxyguanosine combines with monophosphate to form deoxyguanosine monophosphate or DGMP guanosine monophosphate, or it is also known as deoxyguanolic acid. This is a deoxyguanolic acid. The third one, deoxycytidine combines with the phosphate group to form deoxycytidine monophosphate, that is DCMP or deoxycytidalic acid. The fourth one, deoxythymidine combines with the phosphate group to form deoxythymidine monophosphate or DTMP or deoxythymidalic acid. So as a result of this, 4 nucleosides and 4 nucleotides are formed. Okay. deoxyadenosine monophosphate or DAMP or deoxyadenalic acid, deoxyguanosine or deoxyguanosine monophosphate or DGMP or deoxyguanalic acid, deoxythymidine. Nucleotide is deoxythymidine monophosphate, DTMP and deoxythymidalic acid. Deoxycytidine, the nucleotide is deoxycytidine monophosphate, DCMP or deoxycytidalic acid, not guanalic acid, it's a cytidalic acid. So when all these nucleotides join together to form a polynucleotide, several nucleotides link to each other to form polynucleotide chain. Look at here, this is one nucleotide, so this is another link. Like this, several nucleotides join together to form polynucleotide strand. So again, come back to the difference. In case of DNA, you find 2 polynucleotide strain. That's why it's called double stranded. Whereas in case of RNA, you find single strand or single polynucleotide chain. So linkage between nucleotides. Okay. So that takes place. Look at here. 3rd carbon and 5th carbon. 2 phosphoester bond formation takes place. So one between the 5 carbon of pentose sugar and phosphate group. The second one between the 3rd carbon of pentose sugar and the phosphate group of successive nucleotides. 2 phosphoester bond formation takes place. That's why it is called phosphodiester bond. Phosphodiester bond. Look at here. So 2 esterification produce phosphoester bonds. So one between sugar and phosphate, the second bond between phosphoester bond between phosphate and a sugar of successive nucleotides. This is the structure of polynucleotide chain, when many nucleotides join together to form polynucleotide chains. So the structure of polynucleotide chain in case of DNA we'll discuss in the next session.
Thank you.